Thanks very much, everybody. I'm really pleased to be able to talk with you. It took us a long time, actually, to come up with a good face pun that I thought would really approach this topic. So your face is really your window to the world. It's at the core of your identity. It's the way that you interact with others. But have you ever stopped to wonder, how does the face develop? This is a question I'm really interested in. So you have to take this single cell, a fertilized egg, and build this complex three-dimensional structure, right? And that structure has to be able to speak, to breathe, smell, see. And so development has to be stereotyped enough to accomplish all of those functions. But at the same time, there have to be enough differences in craniofacial development to allow us to recognize each other. For example, a newborn baby recognizing her mother. So my lab is really interested in understanding the basic cellular and molecular mechanisms by which the development of the face is controlled. It turns out the development of the face starts from very early stages of development. In fact, the cells that are going to give rise to the face are actually shared with some of the cells that are going to give rise to the brain. So here what I'm showing in this blue picture on the left is a mouse embryo at about 8 days of pregnancy. The head is facing up, the tail is facing down, and we're looking down at the back of the embryo. These sort of wing-like structures, they're not wings. These are mice. These wing-like structures are called neural folds. These neural folds are going to roll up and form a neural tube. That neural tube is going to give rise to the brain and the rest of the central nervous system. But these pink cells are very interesting. They're called neural crest cells, and they're going to take a long journey and ultimately provide the basic raw material to give rise to the face. So here we have a picture of those neural crest cells as they begin that journey. They start to migrate towards the front of the face. Here we're looking at the side of this same embryo. And so in order for these neural crest cells to reach their destination, their migration has to be tightly controlled. And this has to be controlled by cell signaling between those pink cells and the surrounding blue cells. And ultimately, they're going to make it to the front of the face there, where they're going to contribute the bulk of the craniofacial skeleton. In fact, most of the bone, cartilage, and connective tissue of the craniofacial skeleton is derived from these neural crest cells. So by the way, this is a micro CT scan of a mouse head. And if you've ever had a CT scan, the device that we use for these mice is exactly the same as what you're picturing. It's just much smaller. So this is a complex process. And maybe because of this, birth defects of the face are very common. In fact, craniofacial malformations constitute about 1/3 of all birth defects. You probably all know somebody that has a cleft lip or a cleft palate. A cleft lip is just a hole in the front of the upper lip. Cleft palate is just a hole in the roof of the mouth. Another common birth defect is craniosynostosis. This is a disease where the bones of the skull fuse inappropriately. So normally when a baby is born, the bones of the skull have to remain separate, and that accommodates the rapid growth of the early brain. But when a baby has craniosynostosis, these bones fuse, and that can actually squeeze the brain and cause all kinds of brain damage. So to date, prevention of these diseases has not been possible. In fact, there's not a single medicine that we can use as a therapeutic against these types of diseases. So my lab is a part of the program in craniofacial biology at UCSF, a group of scientists and clinicians that are dedicated to understanding and improving treatment of craniofacial conditions. On the clinical side, we have geneticists to help to diagnose these diseases, surgeons and orthodontists to help to repair these defects, and then social workers and speech pathologists to help families deal with the impacts of the ultimate malformation. And even though the surgeries to correct these malformations are quite good, depending on the birth defect, patients can still be left with significant malformation affecting speech and self-esteem. So in a similar way, we've also established a group of basic scientists who are very interested in understanding the basic underlying biology that controls craniofacial development. My lab is very interested in a disease called craniofrontonasal syndrome. This is a disease that only affects females, and these females have a smaller middle of the face. They have wide-set eyes. This is a patient with craniofrontonasal syndrome here. They have this problem with the bones of their head where they're fused incorrectly. And they often have cleft lip and palate. This patient does not have a cleft lip and palate. They also often have abnormalities of the brain. So CFNS is caused by mutations in a gene called EphrinB1. FRNB1 is a molecule that sits on the cell surface, and it signals to other cells. And it helps to tell cells where to go. So we can really think of FRNB1 as part of the cell's GPS. So most of what we know about CFNS we've learned from mice. And although all of this information is extremely valuable, you've probably noticed that there are some basic differences between mouse faces and human faces. So for this reason, we've also wanted a model of a human cellular model for CFNS. And to do this, we've made iPS cells. We've worked together with the craniofacial group who collected just a small bit of skin cells from the forearms of affected individuals. We can take these cells, and by adding just a few genes, we can transform them into induced pluripotent stem cells. We can then take these induced pluripotent stem cells and turn them into any cell of the embryo and ask how dysfunction in FRNB1 affects those cells. And so here what I'm showing are cells from a CFNS patient that have been turned into these early neural fold cells. And what we find is that dysfunction of FRNB1 leads to a disruption of that cell guidance system. And so now when that system goes haywire, these cells are grouped into these large groups of cells, whereas the control cells are nicely intermixed. So this suggests something very interesting about CFNS, which is, as I mentioned, these patients have both craniofacial anomalies as well as brain anomalies. This may explain that fact, so that actually this disruption of the cell's positioning system is occurring in those progenitor cells, those cells that are like these neural fold cells, that are common both to the development of the brain and to the development of the face. It also suggests a possible therapeutic approach. If we could block this aberrant cell segregation, maybe we could provide therapeutic benefit. So we have a lot of challenges we need to overcome in order to approach this. But I think that trying to understand the cellular and molecular basis for these diseases is an important step in order to design medicines to treat them. Thank you.